Hello everyone, my name is Claire Kim and I'm a part of the Casto Lab. I'm excited to share our research on transcriptional regulation of venom gene expression. We worked with snake venom glands, specifically comparing the main and accessory venom glands across 3 species of rattlesnakes in order to explore how glands evolve and diverge functionally over time. So the rationale for why we chose to study this was that snakes produce venom in modified salivary glands, and most research has focused on the main venom gland, because it generates the majority of the venom and it tends to be larger, but many species also have a smaller accessory venom gland that has been relatively understudied. We wanted to understand how these two glands, the main venom gland and the accessory venom gland, differ in their expression of venom genes and underlying regulatory environments. Our main hypothesis was that the accessory venom gland expresses venom genes at a lower level, potentially due to differences in transcription factor activity and pathway regulation. Our figure 1 illustrates our methods in collecting the main venom gland and the accessory venom gland tissues from 3 species, Crotalus cerberus, Crotalus organis, and Crotalus viridis. Using RNA-seq, we generated a high-quality transcriptome from both gland types in matched individuals. We then used computational tools like DE-seq2 and R in order to analyze expression differences, transcription factor correlations, and regulatory pathway enrichment. Our core findings are in the later figures, with figure 2 being a heat map of venom gene expression across species and across gland types. You'll notice that the main venom gland expresses venom genes at a much higher level than the accessory venom glands, with a few exceptions where genes do show higher expression in the accessory gland. In figure 3, we looked at transcription factors that were correlated with venom gene expression. The accessory venom gland tends to have higher expression of transcription factors that are correlated with venom gene repression, while the main venom gland tends to have higher expression of transcription factors that are correlated with venom gene activation. This supports our idea that the lower expression of venom genes in the accessory venom gland could be due to differences in gene regulatory networks. And then figure 4 is our pathway enrichment, in which we perform the enrichment analysis on known pathways like the unfolded protein response, UPR, and extracellular regulated kinase signaling, ERK, both which are important for venom gene activation and regulation, as shown in figure 4a, which was done through previous research. Panel 4b highlights a venom-associated transcription factor network, where the color coding shows which transcription factors are associated with each gland and whether the venom are unregulated in each gland, with the red being higher in the main venom gland and the blue being higher in the accessory venom gland. Panel 4c shows how accessory venom gland also has lower enrichment for both UPR and ERK, which aligns with their reduced venom gene expression, since the main venom gland has much more enrichment compared to the accessory venom gland, thus it produces more venom. In conclusion, there is a clear and consistent differential expression of venom genes between the main venom gland and the accessory venom gland across species. The accessory venom gland also shows higher expression of transcription factor repressors and lower enrichment for activation pathways like UPR and ERK. These findings suggest that the accessory venom gland has a distinct transcriptional environment and potentially plays a complementary or modulatory role in the venom production. To wrap up, our study sheds light on how transcriptional and regulatory differences between 2 venom-producing glands may contribute to the evolution and specialization of venom systems in snakes. Thank you so much for listening.